Good morning, students. Today we are moving on to our next topic, the production, isolation and utilization of the phytoconstituent podophyllotoxin. Podophyllotoxin, the phytoconstituent, it is isolated from resinous material. Resinous material which is called podophyllin, and this resinous material is obtained from the roots and the rhizomes of the plant Podophyllum immodi and Podophyllum peltatum and other species of Podophyllum like Podophyllum hexandrum, etc. And this plant belongs to the Barbaridaceae family, as we have mentioned here. As far as the geographical distribution of this plant is concerned, it is widely distributed in Asia region, in America also, in South America also. It is widely distributed around the world. So if you look at the structure of phytoconstituent podophyllin or podophyllotoxin, this is the structure of podophyllotoxin, and if you see the structure, you will observe there is a lactone ring, there is aromatic alcohol, and there are three methoxy groups, 1, 2 and 3, methoxy and an acetal. Since we are here concerned about the structure or the phytoconstituent alone, these functional groups, they are very important in terms of determining or in terms of predicting the biological activity of the proposed or the phytoconstituent which we are going to isolate. So on the basis of these, you can say these functional groups, we can assume that this compound might be having that particular activity. Actually, podophyllotoxin, it falls under the category of lignans. It is a secondary metabolite. Lignans, they are secondary metabolites, and they are polyhydroxy secondary metabolites. So it is very important to know that podophyllotoxin are secondary metabolites and they are polyhydroxy in nature. So on the basis of structure and on the basis of functional groups, we can predict the activities, and we have found that lignans, they are generally having a strong antioxidant activity, they are having a strong anti-tumor activity, and they are having potent estrogenic activity also. So this is the introduction of podophyllotoxin. Moving on to the physical properties of podophyllotoxin. Podophyllotoxin occurs in the form of amorphous powder, which is of light green to greenish yellow in color. It is soluble in alcohol and other organic solvents, and the solubility is very important because we are going to isolate the compound from the raw material. So it is soluble in alcohol and organic solvents and sparingly or rarely soluble in water. If we look at the production method we follow for production of this podophyllotoxin and its isolation, we follow the simple procedure where what we do, we took the root powder, we extract the root powder with ethanol using Soxhlet apparatus for a period of 6 hours. After the extraction, the extract is concentrated to a syrupy mass. This extraction or this concentration is done in rotary vacuum evaporator. Then to the syrupy liquid or syrupy mass, we add water and dilute HCl. After addition of water and dilute HCl, the mixture is cooled and keep aside for around 2 hours. After 2 hours, the mixture is then filtered, and to the filtrate we do the washings with acidified water. After these washings, the mixture that we obtained is cooled at 5 degrees Celsius or below 5 degrees Celsius, and after cooling the residue is mixed with 95% hot alcohol. The mixture is then filtered and evaporated under rotary vacuum evaporator, and we obtain the light brown powder of Podophyllotoxin after these procedures. So now we have isolated the Podophyllotoxin. We are also matching the color of the Podophyllotoxin with the reported literature. So it is light brown Podophyllotoxin. But in phytochemistry we cannot rely upon the physical property alone. We also have to perform some chemical tests for determination of the exact identity of this the isolated phytoconstruct. So here we have an identification test. This identification test is especially for lignans. This is a special test for lignans. What we do, we take the aqueous solution of the extract which we supposed to contain the Podophyllotoxin, or you can also take the isolated Podophyllotoxin sample, then to which you add 2 ml of 2% furfural and after addition of furfural you will observe the red color, and if you observe the red color, then it shows the conformity test for the presence of Podophyllotoxin and lignans. So moving on to